Okay, let's unpack this. Imagine, just picture the most incredible, intricate, vital transport system you can possibly conceive.
Right.
We're talking about the one inside every single one of us right now. Today, we're taking a deep dive into the human circulatory system.
And what's fascinating here really is that while it seems simple on the surface, a pump and some tubes, its elegance, its genius lies in the design.
Mm-hmm.
It allows for such precise control, adaptability, often in ways we just completely take for granted, until something goes wrong, of course.
Absolutely. And our mission today, it isn't just to skim the surface. We want to truly master this foundational information, drawing from expert physiological sources, really get that professional level view.
Yeah, get into the nuts and bolts.
Exactly. We'll be answering some big questions you might have, like how does blood actually manage to flow in just one direction throughout your whole body?
Good question.
How do the different parts of your blood vessels, how do their structures contribute to their specific jobs? Why does blood pressure change so much as it moves through the system? And why is that important?
Crucial.
And what's the significance of how a total area of your blood vessels changes? It's kind of surprising. So yeah, get ready for some serious aha moments.
Definitely.
So if we're trying to crack the code here, where do we start, I think? Maybe with its core purpose.
That's good.
At its heart, this system, your body's essential delivery service. Simple as that almost.
Well, yes and no. It is responsible for transporting and distributing the essentials like oxygen and nutrients to all your tissues.
Right, the good stuff in.
Exactly. But it's not just delivery, it's also pickup.
Yeah.
It has to remove the metabolic byproducts, the waste.
Okay. So waste removal too. Critical.
And its role stretches far beyond just transport. It's absolutely pivotal in homeostasis.
Homeostasis, maintaining that internal balance, right?
Yeah. Precisely. Your body's incredible ability to keep things stable inside, regardless of what's happening outside. So regulating body temperature, keeping fluid balance right, adjusting oxygen and nutrient supply.
Like when you exercise versus when you're just sitting.
Exactly that. The circulatory system is central to all those adaptive processes. It's constantly adjusting.
Okay, so it's way more than just plumbing. It's like climate control, waste management, delivery, all rolled into one.
That's a good way to put it.
So who are the main players in this operation? Can we break it down?
Yeah, we can think of three main elements. First, the pump, the heart, tireless.
The engine.
Then you have the distributing and collecting tubes and the blood vessels. Think of them as the roads.
Highways and side streets.
Kind of, yeah. And finally, this extensive system of incredibly thin vessels, the capillaries.
Ah, the capillaries. That's where the real action happens, the exchange.
That's exactly where the critical exchange between blood and tissues takes place. It's the destination, in a way.
Okay. Pump, tubes, exchange sites. Makes sense.
And of course, the fluid itself. Blood, filling these vessels, doing the actual carrying. It's a complex, heterogeneous fluid, essential for everything we're talking about.
Right. Can't forget the blood itself.
Yeah.
Okay, so let's talk about the heart. You said it's the pump, but it's not just one pump, is it? It's more complex than that.
That's absolutely right. It's really like two pumps working together in series, one after the other.
Two pumps, okay. How does that work?
Well, the first pump handles the pulmonary circulation. It propels blood through your lungs.
For oxygen, right. Get oxygen in, CO2 out.
Exactly, that vital gas exchange. Then, once the blood is oxygenated, it comes back to the heart, to the second pump.
And that one sends it everywhere else.
Precisely. That handles the systemic circulation. It propels that oxygen-rich blood to all the other tissues in your body. Brain, muscles, organs, everything.
Wow. Okay, a dual pump system.
Yeah.
Very efficient. But how does it make sure the blood only goes one way? You can't have it flowing backwards.
No, definitely not. That would be chaos. Unidirectional flow is achieved by a really clever arrangement of flap valves.
Flap valves, like little gates?
Essentially, yes. Like highly sensitive one-way doors. They open to let blood through in the right direction and then snap shut to prevent any backflow. It's all about timing and pressure differences. Really quite elegant.
Okay, that makes sense. Valves ensure one-way traffic. Now, here's something that always puzzled me. The heart beats, right? It pumps intermittently. Contraction, relaxation.
Right. Systole and diastole.
Yeah. But the blood flow out to my fingers, say, feels pretty continuous, not like spurts. How does that work? How do you get continuous flow from an intermittent pump?
That's a fantastic question. And the answer lies in the physical properties of your large arteries, especially the aorta.
The big one, leaving the heart.
Exactly. It's not just a rigid pipe, it's very elastic. So during systole, when the ventricle contracts and pushes blood out, The aorta and its main branches actually stretch. They distend to accommodate that surge of blood. They swell up a bit.
Okay, they stretch.
Then, during diastole, when the heart relaxes and refills, the stretched elastic walls of these large arteries recoil, they snap back.
And that pushes the blood forward.
Precisely. That elastic recoil maintains pressure and keeps the blood flowing smoothly onwards. Even when the heart itself isn't actively pumping, it dampens the pulsations.
So the arteries act like a pressure reservoir, smoothing out the pulses.
Exactly. It turns that intermittent ejection into a much more continuous flow for the tissues downstream. Really ingenious.
It really is. Okay, what about the total amount of blood? Is it always the same amount in this system?
In a normal, healthy person, yes. The total blood volume is essentially constant. It's a closed system.
So if blood builds up in one place,
it has to decrease somewhere else. The total volume stays the same. But, and this is key, the distribution of that blood is highly dynamic.
meaning where it goes changes.
Constantly. It's precisely controlled. Depends primarily on two things. The output of the left ventricle, how much blood the main pump is pushing out, and the contractile state of the resistance vessels, mostly the arterioles, in different regions.
Arterioles. We'll come back to those, the stopcocks, right?
The very same. They control where the blood flow is directed. More to your muscles when you run, more to your gut after you eat. It's all regulated.
Okay, so the system isn't just one single loop. You mentioned vessels arranged in series and in parallel. What does that mean functionally?
Right, think about it like electrical circuits almost.
Yeah.
Series means blood flows sequentially. Large artery, smaller artery, arteriole, capillary, venule, vein, one after the other.
Okay, like steps in a process.
Yes, exactly. Each step has a different role, different vessel characteristics. Parallel means that blood flow from the arteries splits off to supply different organs or capillary beds simultaneously.
So blood goes to the brain and the kidneys and the legs all at the same time.
Precisely. And the beauty of the parallel arrangement is that you can control the flow to one area, say, increase flow to muscles, without necessarily cutting off flow to, say, the brain. It allows for independent regulation.
Ah, I see. That parallel structure is crucial for directing resources where they're needed most.
Absolutely critical for managing pressure, flow, and meeting variable demands across the body.
Okay, let's zoom in on the vessels themselves. They're not all built the same, are they? You mentioned different characteristics.
Not at all. They're highly specialized. Blood moves super fast through the aorta and the big arteries.
The highways.
Right. But as these branches go further out towards the periphery, they get narrower and their walls get thinner. And importantly, their composition changes.
Composition, like what they're made of.
Exactly. The relative amounts of elastic tissue, smooth muscle, and fibrous tissue, mostly collagen, vary significantly between vessel types.
And that difference in material gives them different properties.
Vastly different properties. Physical properties, physiological properties. It dictates how much they can stretch, how much they can contract, how rigid they are.
Can you give an example? Like the aorta versus something smaller.
Sure. The aorta, coming right off the heart, dealing with high-pressure pulses, is predominantly elastic. That stretch and recoil we talked about. That's the elastic tissue.
Got it. Lots of elastic.
But as you move out to the peripheral arteries, they become more muscular. They have more smooth muscle in their walls.
Why more muscle?
That muscle allows them to actively constrict or dilate to change their diameter. That's crucial for controlling blood flow and pressure further down the line.
Okay, so elasticity near the heart, more muscle further out.
And then you get to the arterioles. There, the muscular layer really dominates. They are highly muscular compared to their size.
Which brings us back to the stopcocks. Why that nickname? Why are arterioles so important?
Well, think about resistance to flow. In the big arteries, there's not much friction, relatively speaking. The pressure doesn't drop much.
Okay. Wide pipes, easy flow.
Right. Small arteries start to add some moderate resistance, but the maximal resistance is found in the arterioles.
The most resistance. Why? Because they're narrow and muscular.
Exactly. They're narrow, and that muscular wall allows them to clamp down significantly or relax and open up. They act like tiny little faucets or stopcocks controlling flow into the capillary beds that follow.
So if they offer the most resistance, that must be where the biggest pressure drop happens.
Precisely. The single greatest pressure drop in the entire circulatory system occurs across the arterioles and the very end of the small arteries leading into them.
Wow. So the pressure entering the capillaries is much lower than arterial pressure.
Much lower. And this pressure drop is essential. It's a direct consequence of that high resistance. And by adjusting the contraction of the smooth muscle in the arterial walls,
They can fine-tune flow.
they can fine-tune blood flow to specific tissues with incredible precision. Open the tap, close the tap. And just as importantly, adjusting that resistance helps control your overall arterial blood pressure.
Amazing. They're like the gatekeepers, controlling local flow and overall pressure.
You could definitely say that. Gatekeepers is a good analogy.
So after the arterioles, we get to the capillaries, the exchange hub.
The business end of the circulation, you might say?
And something interesting happens to the flow there too, right? It stops pulsing.
Yes, another remarkable transformation. That pulsatile flow coming out of the arteries gets damped down. By the time blood reaches the capillaries, the flow is essentially smooth, non-pulsatile.
Oh, is it the arterioles again?
It's a combination. It's partly the elasticity of the large arteries absorbing the initial pulse, as we discussed. But it's also very much due to the high resistance offered by the small arteries, and especially the arterioles. That resistance acts like a filter, smoothing out the pressure waves.
So the resistance doesn't just drop the pressure, it smooths the flow too.
Exactly. Which is perfect for the capillaries because you want a steady, gentle flow for efficient exchange, not hammering pulses.
Makes sense. And the capillary network itself, it's enormous, right?
VAST doesn't even begin to cover it. Estimates are around 10 billion capillaries in the human body. 10 billion!
That's astronomical.
And many arise from every single arteriole. Now, here's the fascinating part. Each individual capillary is incredibly tiny, narrower than a hair.
Right. Super small cross-section.
But if you add up the cross-sectional area of all those billions of capillaries together,
The total area must be huge.
immense. The total cross-sectional area of the capillary bed dwarfs the cross-sectional area of the aorta. It's orders of magnitude larger.
Wow. Okay, so tiny individual pipes, but a massive total width for the river, so to speak.
Exactly. And think about what happens to flow velocity when a river widens
It
dramatically.
really goes down, right?
Precisely. Because of that enormous total cross-sectional area, the velocity of blood flow becomes very, very slow in the capillaries.
And that's why exchange can happen effectively, because the blood is moving slowly.
That's a huge part of it. Slow velocity gives time for diffusion. Combine that with the fact that capillaries are short tubes with walls only 1 cell thick.
Super thin walls.
And you have the absolute perfect conditions for rapid, efficient exchange of oxygen, CO2, nutrients, waste products between the blood and the tissues. It's all about maximizing surface area and time.
So it's an inverse relationship then, where the total cross-sectional area is small, like the aorta, velocity is high. Where the total area is massive, like the capillaries.
Velocity is very low. You got it. From aorta to capillaries, the area increases maybe 500-fold, and the velocity drops proportionally. They're like mirror images on a graph.
A beautiful example of structure perfectly matching function.
Couldn't have said it better myself.
Okay, so blood flows slowly through the capillaries, does its exchange job. Then where does it go? And how does volume distribution look across the whole system? Where is all this blood actually sitting at any given moment?
Right. After the capillaries, blood collects into venules, which are small veins. Then these merge into larger and larger veins, eventually returning to the heart.
And what happens to the cross-sectional area and velocity now?
As the venules merge into veins, the total cross-sectional area starts to decrease again compared to the capillaries, but it's still larger than on the arterial side.
Okay, so area goes down from the capillary peak.
Which means the velocity starts to increase again. Blood speeds up as it heads back towards the heart, but not as fast as in the aorta.
Okay. Now, the distribution numbers. You mentioned about 5 liters total blood volume for a typical adult.
Roughly, yeah. Around 5,000 milliliter for a 70-kilogram woman as a reference point.
And where is most of it?
Okay, get this. About 84%, so 4,200 milliliter, is in the systemic circulation, the main body circuit.
Okay, the vast majority.
But within that systemic circulation, look at the breakdown. Aorta and large arteries, only 300 milliliter, 6%. Small arteries, 400 milliliter, 8%. Capillaries, just 300 milliliter, another 6%.
So arteries and capillaries hold relatively little at any one time.
Exactly. Now here's the kicker. Small veins, 2,300 milliliter, that's 46% of your total blood volume.
Wow. Almost half.
And large veins hold another 900 millioel, or 18%. Added up, the systemic veins and venules contain about 64%, nearly two-thirds, of all your blood.
So the veins are the main reservoir.
Absolutely. They're often called capacitance vessels. They hold the bulk of the blood volume, acting as a reservoir that can be mobilized if needed, like during blood loss or exercise.
That's incredible. So it's not just about return, it's about storage.
Very much so.
What about the lungs and the heart?
The pulmonary circulation holds much less, around 440 milliliters, or nearly 9%.
Yeah.
Pretty evenly spread across its arteries, capillaries, and veins. And the heart itself, when it's full just before beating end diastole, holds about 360 milliliters, or just over 7%.
Fascinating breakdown. And one last point on the return trip, the venae cavae, the big veins returning to the heart. How do they compare to the aorta?
Interestingly, their total cross-sectional area is actually larger than the aorta's.
Really? Even though they're veins?
Yes, and because area is larger, the flow velocity in the venae cavae is slower than in the aorta.
Ah, so slower return flow into the heart. Makes sense for a low-pressure system.
Exactly. It allows for smooth filling of the heart without turbulence.
Okay, this deep understanding of normal flow, pressure, resistance, volume.
Yeah.
It's not just academic, is it? It's crucial for understanding disease.
Absolutely. Clinical correlations are where this knowledge really comes alive.
You mentioned an example, hyperthyroidism.
Right, like in Graves' disease. This is a condition where your basal metabolism is significantly elevated. Your body's basically running in overdrive all the time.
Burning more fuel, needing more oxygen.
Precisely. And how does the body respond to that increased tissue demand? Often with arterial or vasodilation.
Those stopcocks again. They open up. Dilate.
They dilate. They relax their muscular walls to allow more blood flow to the metabolically active tissues.
Okay, makes sense. More demand, open the taps wider. But what's the consequence related to what we discussed about damping?
Well, remember how crucial the resistance in the arterioles is for damping those pressure pulses from the heart?
Yeah, smoothing the flow for the capillaries.
When the arterioles vasodilate significantly, their resistance drops. And when resistance drops, their ability to dampen those pulses diminishes.
So the pulse pressure gets through more easily.
Exactly. The pulsatile nature of arterial pressure isn't smoothed out as effectively. And you can sometimes actually observe pulsatile flow in the capillaries in these patients.
Pulsing capillaries. But you said that's not normal. Steady flow is ideal for exchange.
It's definitely not ideal. And seeing that clinically really underscores the vital role arterioles play, not just in controlling how much flow goes where, but also in maintaining that steady, non-pulsatile environment crucial for capillary function. It highlights their dual role, resistance and damping.
Wow. That's a powerful clinical link. Okay, so let's try and wrap this up. What does this all mean for you listening right now? We've gone deep.
We really have.
From the heart's amazing dual pump system, the clever flap valves ensuring one-way flow, to how blood vessels are specifically built for their jobs, elastic arteries, muscular arterioles.
We saw how pressure plummets across those crucial arterial stopcocks and how that controls flow.
And the incredible design of the capillary network. Huge area, slow flow, perfect for exchange, plus where all your blood actually hangs out most of the time, mostly in your veins.
It gives you that professional level grasp of the fundamentals.
Yeah, you now understand how velocity and area are inversely related, how volume is distributed. So looking at the big picture.
The big picture is that the body isn't just parts. It's an integrated system. Structure is exquisitely tied to function everywhere you look in circulation.
That balance.
Yeah.
Resistance, elasticity, volume distribution.
It's what allows us to adapt, to go from rest to exercise, to digest food, to manage temperature, all seamlessly. It's dynamic regulation at its finest.
So you've just gained a really deep, almost professional level insight into this fundamental system.
Yeah.
For a final thought to take away, just consider this. How incredible is it that this system, designed for constant flow and delivery, can so precisely tweak distribution, pressure, resistance, adapting minute by minute to everything your body demands, often without you having any conscious awareness of it.
It really is awe-inspiring when you break it down.
What stands out most to you about the sheer ingenuity, the cleverness, let's say, of the circulatory system's design? That's something to mull over until our next deep dive.